Steve Osofsky. I'm a professor of wildlife health and health policy and director of the Cornell K. Lee Siang Center for Wildlife Health. And I'm really excited now to introduce the reason you're all here. It's Dr. Pete Coppolillo. Pete and I were partners in crime back in the day at the Wildlife Conservation Society. I remember wandering around Tanzania with you, Pete. I'm just going to read Pete's bio. Pete received his bachelor's degree with an honors in biology and environmental conservation from the University of Colorado and a Ph.D. in ecology from the University of California, Davis. Pete worked with WCS, the Wildlife Conservation Society, for 10 years, first at their New York headquarters and then as part of the Africa and North American programs. He studied ferruginous hawks in North America, avian community ecology in Kenya, and large herbivore ecology and herding systems in Tanzania. He has helped plan and carry out conservation strategies in Bolivia, Ecuador, Argentina, Congo, Cambodia, Tanzania, as well as in the U.S. His publications have appeared in conservation biology, biological conservation, Public Library of Science, human ecology, landscape and urban planning, landscape ecology, and science. And he's the co-author of the book, Conservation, Linking Ecology, Economics, and Culture. So thank you so much for coming, and let's welcome Dr. Coppolillo.
Thank you all for squeezing this in on your lunch breaks, and thank you, Steve, for having me. You get special thanks because that's what Steve looked like when I met him, the young buck. But we're a now 30-year-old organization, but we're just off in Montana doing our own thing. So it's a great opportunity for me to get to come to a place like Cornell to it's just, kind of mind blown all day long. And so it's really meaningful for me personally, but for the whole organization, to drum up new projects and get new ideas and do better work. So this is kind of us in one slide. And really I'll tell you a little bit about, we also have 4 pillars. I'll tell you a little bit about each of them, but really I'll go into more detail about those tomorrow night. Today I'm really just going to focus on our biosecurity work because it's the newest part of what we do. But before I go there, I'll just cover some of these. We're conservation dogs are a relatively new field. When I started, we had 6 active dogs, and we've got almost 100 now. And we've worked in a lot of places all over the world. So it's increasingly sort of mainstream kind of stuff. But our organization we used to do all the work ourselves. We'd take our dogs and go out to the field and do the work. And now we do a lot more consulting. We still do a lot of project field work where it's our dogs and they come and go and do the thing. But a lot of our growth has been fueled by capacity building. So, 61 of those 98 dogs are on the ground in Africa, and they're handled by African handlers who either work for NGOs or host country governments and stuff like that. So we work in a lot of different ways and in a lot of different contexts. And that's why it's so valuable for me to be here is to just to get to learn what's happening on the ground, where we need it, what's new, what other new applications could dogs help with. One other thing I'll mention about us as an organization is that we're a little different than a lot of working dog institutions in that we rescue the dogs that we train. I'll talk a little bit more about that tomorrow night, but that's for some practical reasons, and it's also for ethical reasons. But it does introduce a different dimension to our work because they're not all the same. Every one of them is different, and we screen about 1,000 dogs for every one that we actually take. So that creates some differences in terms of how we work and how quickly things move or don't move forward. I'll also mention that I am not a dog trainer. I'm a conservation biologist by training, as Steve's the bio sort of mentioned. My dog experience is mostly bird hunting in Montana with my own dogs, so I get a lot of free dog advice from my staff and everything. But I really came to this work from the conservation side. I was just sold on all the different things that the dogs could do. Many of you, particularly from the Behavior Institute, will be much more experienced, much better dog trainers than I am, and may know a lot more about all that. But I glean, what I can and what I need to here and there. But we have a fabulous staff. So if you guys have questions I can't answer, I will pass them on. But please, as I go through this, stop me if I'm not being clear or if I'm using terminology that's different. One of the things I've learned about the dog training world is everybody's got their own perspectives on it. So I'm here to learn and to make sure that I'm communicating clearly. So if you guys want to stop me or ask questions or get feedback along the way, fire away. I don't mind. So our field really comes from grows out of conservation biology. And many of you may know species like these. These are all species that people can work on for years at a time and never actually see. And in conservation, of course, we're often concerned with things that are declining or rare or hard to find. And so if somebody comes to me and says, how do we protect wolverines? And I don't know where they live or what they're eating or what's eating them or what's killing them, whether it's a pathogen or another animal, it's very difficult to say, here's what you have to do to conserve them. And so as conservationists, we tend to often ramp up, capture and put more collars out and do these things. And what we've learned is, particularly as technology and laboratory techniques have gotten better, we're able to do a lot non-invasively. And so our field was really born in the 90s when it became possible to extract DNA from scat, from poop, right? And so our dogs, for a lot of our history, have worked a lot like you might see a bird dog working. So usually when we're doing ecological monitoring projects, a handler will walk a transect and the dog will quarter back and forth, sampling that sort of area, looking for whatever the target is. Often it's scat, right, for grizzly bears or lion or leopard or cheetah or moon bears in China, whatever it is. But what's interesting about this process is, number one, the dog runs a lot. They go back and forth. They cover a lot more ground than we do. But they're also sampling a much larger area because of this superpower they have that is olfaction, right? So odor could be coming from up to 600 meters away. We've had a dog on a transect who made a right turn and ran 600 meters to find a cheetah scat. So they're sampling a much larger area than we do. So they're really good at that. And nowadays we're seeing a lot more conservation dogs in the literature, whether it's popular literature or the scientific literature. So when I started doing this job 13 years ago, most of the places I went, people had never heard of a conservation dog. And now it's a little bit more common and a little bit more mainstream. And part of the reason for that is they're really good at what they do. I'll show you some more data. If any of you make it tomorrow night, I'll show you more data that I find actually more interesting than this. But this was from Grimm and Seyfarth, a recent review of conservation dogs. This is just detection. But in 89% of the cases reviewed, the dogs were better than the conventional model. Like I said, that's not that interesting because if you're resorting to dogs, you've probably exhausted a lot of the conventional models, or the standard inexpensive way to do it isn't effective or isn't useful for you. So this is not to say everyone should be using dogs, but when people do choose to use them, they're often good at it. We call this the greatest hits slide. This is some of the stuff that dogs have done, and this is compiled over probably 20 years of work, but we're always adding new things. This was just 2 years ago in the Falkland Islands. It was a Shiba Inu, which is not a common working dog breed, but he found a single rat on a boat, and in fact, I put that as more than 60 times because there were 5 humans on the boat who also were searching. They never found it. It was an experimentally placed rat. And they never found it. So there are just certain things that dogs can do that we simply can't do. So they open a lot of doors for us. And you guys probably know this story well. It's because of their superpower, is olfaction. It's not hyperbole to say a dog's nose really is the best chemical sensor the world has ever known. And that's true for a number of reasons. One is they are so sensitive. We're down to the single digits in parts per trillion now that people demonstrate dogs can detect, and that is amazing. But really, the most amazing part of a dog's nose is that it's attached to a dog, right? They love us. They want to work for us. They'll go to the forest. They'll jump over logs. They'll go all sorts of places. And we can train them on a new odor in an afternoon. So they're incredibly flexible. And that's really, and they want to work with us as humans because we co-evolve. So that's what's really powerful. But I want to also mention one other thing that we're going to talk a little bit about today, which is dogs have another superpower that lots of people aren't aware of. The low threshold, the low odor concentrations is lots of people know about that. And that's what we use for bombs and drugs and all sorts of stuff. But dogs can also tell the difference between two very similar odors. And we call that discrimination. And that's the superpower that I'm going to focus on primarily for the talk today, because that's what we use for disease and for One Health sort of work. And so, the analogy that a lot of the dog folks will tell us is you and I walk down the street and we smell bread baking. And we say, oh, that's bread. But a dog will smell all the components of that bread, the flour, the sugars that are developing, the yeast, the residue in the oven, all of it. A dog will recognize all of those different things and tell the difference between that loaf of bread and a different loaf of bread even. So that's a cool and important distinction. And I'm going to focus on our disease work. And there are two types of disease work that we do. One is just we collect all the scats we can find or all the samples we can find from a species, and then we send them to the lab. The dog is just collecting scats at that point. They're not discriminating. They're not telling the difference between any of them. So those are interesting. That's important. It's important work. Sometimes that's the kind of work that we need to do. I'm not going to spend a lot of time talking about that. The other one is when the dog is doing the discrimination. The dog is telling us this is a sample. This is from an individual that is infected with something. And so that we're doing that kind of work. We did the original for that. Sorry. We did the original work on that with brucellosis. I live in Bozeman, Montana, where our elk and our bison have brucellosis. But we're also doing it with chronic wasting disease. But what I'm really going to talk about mostly today is Mycoplasma ovipneumoniae in bighorn sheep. And the reason for that is because we've dived really deeply into that project and realized that we're in the deep water. And that's why I'm here, is to talk to Steve and to talk to all of you and other colleagues here about how we can do this more deeply, more confidently, and really understand what we're learning from the dogs and from and what a dog's alert means. So bighorn sheep are common in the West, but they're a lot less common than they used to be. So in 1850, the Lewis and Clark journals, they were one of the most common ungulates on the prairies. And in 1960, they hit their low point, and a big driver, not all of it, but a big driver. I know, a big driver from 1850 to 1960 was Mycoplasma and the disease that is still a huge issue for bighorn sheep. Domestic sheep can have it. It's not a serious infection for them. There is a small dip in productivity for them, but it's catastrophic for bighorn sheep. Whole herd, all age die-offs. And so it was Mycoplasma that got us from here to here, and basically it's helicopters that have gotten us from here to here. Because virtually every state in the West, Canadian provinces, tribal wildlife management authorities, everybody's trying to restore their sheep populations. And they do that by moving sheep around. Introducing populations in areas where they're secure, where they're away from domestic sheep, and put them back into mountain ranges. And this is how it happens. It's intensive sort of work. That's a drop net. So they'll bait and the sheep will eat under there for days and days at a time, and then all of a sudden when they've got a lot of them under there, they'll drop the net, or they net gun from helicopters. And then when they get a bunch, they bring them back and they're all processed. So those sheep might be moving to start a new population, or they might be just managed. They might just be putting collars on them, or now increasingly what a lot of agencies will do is what we call test and remove, taking the diseased animals out of the population so that they don't infect others to avoid these catastrophic sort of die-offs. So in all of these cases, no matter when you're handling sheep, you don't want to be moving diseased animals around, right? Because we don't want to compromise the population they're going to. Or if you invest all that time and money, $1,000 an hour to fly around in a helicopter, you don't want to put a bunch of sheep in a new mountain range if they're just going to go over there and die. So it's a big intense operation. These capture operations, there are hundreds of volunteers. It's chaos. It's high stress because nobody wants to lose an animal. And they have to process them quickly and get them out the door. So, and disease screening is part of this process. But here's the problem. The disease, the samples, when you've got sheep in hand, we don't want to put all of them together. So what happens is they catch a bunch of sheep, and then they put them in stock trailers, right? They take the blood or the nasal swabs. I'll show you the samples a little later. And then we send them off to Waddle, to Washington State, to be analyzed. So, and often sheep captures will happen on Mondays and Tuesdays or Sundays and Mondays because they need to FedEx it to Waddle to get the results back before the weekend. So you remember that helicopter costing $1,000 an hour and the helicopter pilot who wants to make $1,000 an hour? Everything stops while we sit there and wait for Waddle to give us an answer. We had a capture in Texas just recently where a rich oil man who had a Learjet said, "I'll just fly him to Washington for you." We said, "Great, you want to wait around for the results?" And they did. But often what happens is the biologist goes and climbs the hill and waits for a text message, and it says, number 17 is positive, number 15 is negative, and all of that. So this is a hugely inefficient way to do a very intensive sort of process. So the Wild Sheep Foundation came to us. Oh, I should also mention, there are some emerging technologies. Many of you have probably heard about Biomeme. That's one of our wonderful collaborators, Kate Hivert, who's at Washington State University, running a Biomeme in a rental car at a sheep capture in Nebraska. And LAMP and RPA are two new versions of basically PCR in the field to do this. So we're also using dogs in conjunction with these other sorts of methods. But we're all trying to figure out a faster, more accurate, more efficient way to do this so that we can continue and help our agency partners conserve sheep and restore sheep to all these different populations. So the Wild Sheep Foundation came to us and they said, "Hey, can you guys help us do real-time animal side testing? When we've got animals in hand, can you have a dog tell us, is this animal infected or not?" And the second part, which I'm really committed to, managers are, they vary on what parts of these are interested in. I want to do this non-invasively. I want to be able to do it without having to capture an animal, right? And we want to do that from scat, because what that means is I can go into a habitat where domestic sheep have been and pick up scat and know, has it crossed over into the bighorn sheep? Because then those sheep shouldn't be a source population for a reintroduction, or they may be in management, or they're maybe on the verge of having a die-off. I'm not going to talk about it. I'm only going to talk about the first two. We also do some work with guardian dogs. This is how it happens. This is how disease happens. You see that guy? That's a bighorn ram. And he probably got the stuffing beat out of him, and he doesn't get to mate with his wild sheep. And then just down the mountain here are all these domestic sheep, which are pretty closely related, and they all come into estrus all at the same time. And he says, "Well, I'll go down there and see what I can do." And often that's how disease transmission happens. So, and so we're working with livestock guardian dogs. Mostly that dog's there to keep bears and mountain lions and wolves away from the sheep. We're working to figure out, can we also use them to keep rams and other wild sheep out of domestic sheep? I'm not going to talk too much about that, but that's part of this whole effort. So all of this started during the pandemic. And we actually got a grant from the state of Montana to evaluate whether we could detect COVID in frontline medical workers' masks. And we said, well, we're not allowed to handle COVID right now. We don't, this was really early days. We don't know what is the right safe handling and all that. Can we use Mycoplasma as a model disease? And if we can detect a respiratory disease in these guys, then explore it more deeply with COVID. And this was right when the sheeple and masks and all the politics, and we're in Montana, so we got a lot of chuckles out of putting surgical masks on sheep, but we did it. And I'll show you the data from all of that. But here's how it works. This is a little different. This is not walking around out in the field. You'll get to see some dogs actually work in this way. But what we do is we take positive samples, training samples that we know are positive. We also take negative samples. We call those distractor odors, and we train the dogs. And when the dog encounters the positive one, they get a reward. And very quickly, particularly with our veteran dogs, they know that this is the game. They know that what they're doing. They get rewarded for that, and they say, okay, that's my target. That's what I'm going to alert to. And as soon as we test them, and as soon as they get up around 80 to 100% in terms of accuracy in the training, then we say, okay, now it's time to test. And we do a double-blind testing because there is skepticism and because we want to know that we're not cueing them. Many of you know the story of Clever Hans. You guys ever heard of Clever Hans? He was the horse who did math. Right? Well, he didn't do math. What he did was he read body language really well. So they'd ask him a question, he'd stamp his foot until he got to the answer, and then everybody would look at his foot, and then as soon as he got to the answer, they'd look up at him and his handler. Well, he just knew to stop stamping when everybody looked up, right? Well, dogs are even better at reading human body language than humans are. So in order to avoid that, we have to do double-blind testing. Dogs will also cheat. Dogs like to get paid just like we do, right? And so we can't use the same samples that we train with for testing because they can just memorize them, right? So we want to know, are you actually generalizing this so that when I show you a sheep you've never encountered, will you actually tell me if it's infected or not? And then just a little word on the terminology here. Police and military guys and will often use the term trained final response. We use the term alert. We do what's called a passive alert. The dog doesn't scratch or bark or touch the sample. They just sit when they see it. So these are all, I'm going to use all those interchangeably. I usually say alert, but they're the dog telling us that's it. There it is. Okay, so I'm just going to summarize some of our initial results for you. And like I mentioned, we did a lot of different things. We did masks, and we'd autoclave the masks and then test with those. That's sheep scat pellets. These are nasal swabs, and we also did cultured MOV. And the long story. Oh, sorry. We also, we used sensitivity and specificity, and then we combine them into a single measure that we call accuracy, and that's how we come up with it. It's just the number of right answers over the number of wrong answers, calculated. So we can give a single measure to say this is how they're doing. And the upshot is they're quite good at it. Using the scat, we got up to 84% accuracy. Some individuals on sheep dung, experimentally infected domestic sheep, were as high as 100%. They were fabulous. They're really good at it. When we used the human nasal swabs, and we also did the medium from the tubes where the nasal swabs were, they also did really well. And same with cultures. So they can regardless of what sample they're using, they were really good at it. So we're feeling really great. We're super cool here. A lot of this work we started back when we were using scent wheels. And scent wheels are fabulous. Our friends at Penn Vet, who you guys have probably heard of, all the cancer dogs, they used scent wheels a lot. Some of them used 12-quart scent wheels, and we had an 8 and a 6. And they're great, and we would put it in an observation room with a camera there, and we could watch them do it, so you can sit outside, so you're not cueing the dog and score all that. It was great. It was great work, but they're a pain to use because after every single rep, you have to take the can out, change the samples. All the cans have to go and get washed. It takes forever, and then you rotate the things around so that it's always in a different spot. It takes a long time. So now we use a gadget called an olfactometer. Super cool. These things have changed. My staff are so much happier now that we have olfactometers because what they do is they've got up to 6 different samples in them at any one time, and they puff the air out of that little hole. So what you see here, this is Leo. You're going to see Leo work here in a second. Leo is he's giving his alert there, and his alert is to sit and to stay there and keep his nose on the source target. And what you see right there are 2 little optical sensors, and there's a beam that goes between those, so the computer knows when he's got his head in the hole, and when he's saying, either he's sniffing it quickly and then he moves on to the next one, or when he alerts, he holds it there, and he's going to keep it there, and then the computer says, yep, you're right, or no, you missed, you're wrong. So you'll see them work. These are actually built from aquarium bubblers and coded. It's still early technology, so it's a lot of DIY, but they're fabulous. They really speed things up and do amazing things. So let's see if I can get Leo to work for you guys. Oops. I have to click on that. There we go. So there he is checking the first one. He didn't even need to go to the second one. He said, it's there. And you hear the beep? That's a treat and train. If any of you know what a treat and train is, it's a remote feeder. And then when it's correct, the treat and train goes, he goes out and he gets his treat. What's happening right now, and this is real time, is each of these machines is puffing air through the tubes to clear it out. And it's getting ready for the next rep. Here comes Leo ready for the next rep. So instead of changing all the cans, we can just go, and there he is, he's giving his alert. And you see the wiggle, he knows when he's right. And he can't sit still, but he keeps his nose in the hole, so it counts. It's all good. Okay, so on to the sheep work. I'm gonna break it down. This has been sprawling, a big project, and I'm gonna break it down into the different components. And the first one is the nasal swab. So during the captures, what happens is animals captured. It's usually just hobbled with so no drugs, but it may sometimes every agency does it differently. Everybody's got their own secret sauce and their way to do it. But what happens is they get swabbed. Nasal swabs looking for Mycoplasma in the nasal cavity. I'm going to refer to swabs. I'm going to try to say swabs, or if I say PCR positive, they're using PCR to get Mycoplasma from the swabs. Or if I'll sometimes say MOV positive, that's in contrast to ELISA positive from a blood test, right? So, if I'm not clear about the terminology. But here's the kicker with these is the first swab goes to Waddle. The second swab gets banked, right? Sometimes we get the third swab, but sometimes there's another project or another something for it, or sometimes they'll send multiple samples in to test. So sometimes we're getting the third or the fourth or the fifth swab out of a sheep's nose. And by the time you get to the fifth swab, there's not a lot of snot left, I'm afraid. So that's a challenge. Keep that one in mind. And then what we do is we bring them back, and those samples or the medium from it goes into the olfactometers, and that's how we test. And what that's enabled us to do is we can not only test more quickly, but run a lot of dogs through the same samples, and test a lot more negatives. And I'll show you some of the other wiggles that we've done in order to figure it out. So we started this work, and we were thinking, this is great. We nailed it on the proof of concept. They had great results. And then we got into the lab, and Leo, by the way, he's the star of all of this. He's the best at this. And we brought the dogs in, we started training, and they were all over the place. It was incredibly frustrating. Frustrating for the dogs, and I will share with you, frustrating for our trainers. There were lots of tears in the lab for this, because they were just everywhere. And we said, what is going on here? And this is what was going on. So you guys probably understand CT values a lot better than I do, but more CT values means it took more cycles to amplify the DNA to get enough DNA to say, yes, we've got MOV here, right? So these samples had a lot of MOV in them. It was great. Easy for the dogs to smell. These samples had much, much less in them, right? And in fact, we had a lot of swabs from animals that came back positive from Waddle, from the first swab, that we had no DNA on. We couldn't get any DNA from them. So that was a big challenge. And that really threw the dogs for a big curveball. It was very difficult. And in fact, what we learned was, remember the training process when I say they get rewarded for smelling a positive swab? Well, those swabs had no MOV on them. So we were inadvertently training dogs on both positive and negative swabs. It was really hard. So one of the things that we did is we said, okay, we've got to simulate swabs. So we're going to culture the strains of MOV from these individual populations of sheep, and then we're going to spike the swabs with the cultured MOV. But because they vary so widely, this is something we want to be able to do in the field at a capture. So what we need to do is we need to mimic the amount of odor that's there, the odor picture for the dog that they're going to encounter in the field. So what we need to do is dilute it. And so working with fancy chemistry and math and our collaborators in the field, what we learned is some of it we can dilute it just 1 to 10, but most of them we're diluting it 1 to 100 or even 1 to 1,000. So these dogs are encountering 1/1000th of the concentration of the original, the first swab. So now we've just gone from a discrimination problem, telling the difference between two things, and we've added a detection problem because now they're at a very low threshold. So it's a hard target for them. And you guys probably understand it even better than I do. We're actually working on the medium that goes with it so that we can dilute it, and that's what goes into the olfactometers, and that's what the dogs respond to. So, but like I said, it allowed us to do a lot more reps and to learn a lot more from it. So we've had 5 dogs on this project. I think that's probably, I don't think we've ever had another project where we've had 5 dogs at once working on it. They train. We worked all the way up to the dilutions, and then worked on both the full strength, and then all the way down to 1,000. And what you see here is at every time they do great, and then we go down to 1 to 10, and they go down. And then they start to do great again. We introduce 1 to 100. They go back down. Zoe had a really rough day. And in fact, Zoe's a rescue from Houston after Hurricane Harvey. She's a street dog in Houston. She's been a great dog for us, but she's getting a little older. This is what inspired Zoe's retirement. Zoe's retired now, I'm afraid. But the other dogs, Leo and Stella and Fenton, they're back on. Same thing happened. We go to 1,000, but then they get back up. And then we introduce a mix. So then it's curveballs. It's, sometimes it's a really strong positive, sometimes it's a negative, sometimes it's, one population or another. So they wobble, but they always come back up. So we're learning a lot about not only how they're approaching, what the disease is and how they're good at it, but how they're approaching these problems and how we can train them to do it better and to be more effective at it. And they're getting much, much better at it. They're really good at it. Leo's the rock star. And the best part about Leo is, poor little Leo, he doesn't get to go to the field. There's just too much in the world for him. He's a German short hairer. He's into birds. He's into anything that moves. So Leo works in the lab. So it's so great that Leo is the rock star of this whole project. He was supposed to be a TSA dog, and he didn't conform to their training specifications. He was at Lackland Air Force Base, and so then they retired him and called us and said, hey, he might work as a conservation dog, and he's been fabulous ever since. But what you can see is there is some variation there. And what we're trying to do is get everybody up to Leo's level here. And so it's a difficult target, and it's a difficult target in 2 different ways. And that's a real challenge. And we've got very small sample sizes. We're only dealing with 34 positive scats that we've gotten from wild captured sheep. When we deal with experimental infections in domestic sheep, we can have as many as we want. But we've got very small sample sizes. And remember, we have to split them from training and testing. So that's another big challenge. We're continuing here to push on. But what we're really trying to do is build up the number of samples that we can get and now confirm them before it ever goes under a dog's nose. We need to know that there's actually MOV there, that it's actually from a positive animal. So on the scat side of things, this is the side that has more potential because there's a lot more poop out there, right? You can walk around and find sheep are herbivores. They're pooping a lot. But it also it has its own set of complications. So at first when we started, we said, well, okay, we trained a lot on domestic sheep. They were good at that. Can we just transition them to wild? Maybe they'll just start recognizing it. Not really. So what you can see there, the sensitivity was only 50%. When they're alerting, they're usually right, but it wasn't great. So we said it's a different odor profile. It's a different scent picture that they're seeing in the bighorn scats. As we move forward, they nail it. They were great when the nasal swab and the ELISA agree, right? So when they're positive on both of them or negative on both of them, As the Brits would say, Bob's your uncle. But they nail it. They're great at it. But when they don't agree, they're just basically guessing. So what's going on here? And one of our collaborators said, well, let me see if there's any MOV in the scat. And long story short, there's not. So we had a dog, we trained Finn. She was a chocolate lab. We trained Finn on culture, and then we gave her scats from positive bighorn sheep. Zero recognition, just nothing. No recognition of it at all. That's when she PCR'd the scats. There's no MOV in the scats. So she said, it's got to be a gut microbiome shift, or there's something else that's. So I'm going to, we'll talk a little bit about the microbiome, but here's a little summary of it. They didn't go straight. We can't go across species. That's unfortunate because it means we can't train on domestic in order to go to wild. Got to stick with wild. And we've got to figure out this challenge for what's going on when they don't agree. And again, I mentioned this a few times. It's hard to go because we got to split the samples. So I'm going to show you a little more about the microbiome. We did microbiome analyses from the nasal swabs, from buccal swabs, and from scat. I'm not going to talk about the buccal swabs, just the nasal and the scat. Again, I'm using the same terminology. And these are funny figures if you're not used to. You guys have probably looked at a lot more of these than I have. But you remember those little plastic plants that all come from one little point? That's what these remind me of. That's what I'm thinking of. So this is a nasal swab, and comparing an animal that is PCR swab positive and ELISA positive with a group of animals that were PCR negative, ELISA negative. Those are the colored dots. The gray ones are ones that were indeterminate, that they couldn't get one or the other from. But as you can see, they kind of group together. So they're quite different. They're different. Statistically, they tell us they're different. You can just look at them, see them, that they're different. And what's going on here is when they're the positive nasal microbiome is much less diverse than a negative animal. So a healthy animal's got a much more diverse nasal microbiome than a positive animal. And for those of you who know and understand all of those things, that's what's going on. Those are all the ones that are there. The fecal story is a little more complicated. So the statisticians tell us these are different, and his explanation to me was, well, you see how the negative ones are in a ring outside, and the positive ones are clustered in. That's the diversity you're looking at. They're different in a lot of different ways. I said, okay, I'll take your word for it. But when we start looking at a whole bunch, on the top, these are the fecal microbiome, and on the bottom, these are the nasal swabs. When we start looking at a whole bunch of different ones, where we have, when PCR, the nasal swab, and the ELISA don't agree, it becomes a lot more complicated. So the red ones are the ones that are significantly different, but I don't, and maybe you guys can help us think about interpreting what these actually mean. And in fact, even right down to the point when we have an animal that's negative on a swab but positive on ELISA. That could be an animal that was exposed at another time in its life, right? It may have had an acute infection and recovered, or maybe it was just exposed and never got acutely ill. We don't actually know. We don't know that animal's sort of history. But those are going to be different than negative sort of ones. So what we realized, and I'm also reassured to hear a few stories here about, well, we just run, we slide them in. We got these guys at Montana State to just run these along with a bunch of other stuff that they were actually doing. And so now we've actually got to go get a pot of money to do this with big samples and figure out what actually is really going on here. So these are relatively small, numbers, 39, 12, and whatnot. But we've got to spend a little time figuring this out. Because when a dog alerts, if that's a manager who's doing a test and remove project, I want to be able to tell him this is what it means, right? So if a dog is alerting and says, some of them, every manager uses a different standard, but some of them will remove that animal, which means kill the animal right now. We're working on trying to get them into captivity so that we can get samples from them. But we want to know what that actually means. Is the fecal shift in their microbiome the same as an ELISA positive animal? We just really don't know. I'm sorry, yeah. No, it's antibodies. They have to seroconvert. It's how Waddle does it for all of them. Yeah. So this is just a little sort of summary. You know, they're great when everything agrees, but when they don't, it's hard to know what's going on. So this is where we're going to spend some time figuring that out. And in fact, one of your alumni here, Danielle Butke from the National Park Service now, has approached us and says, we want to do this for bison. And they've got thousands of samples from bison. And I said, this is the project that the sheep project wants to be. So we're probably going to move these two forward in parallel at the same time to see if we can do a better job. This is what stresses me out about this. This is from a paper. Many of you guys may know this paper. Every one of these is a different respiratory pathogen. MOV is just one of all of these. So I want to know, and I think they are. I think they are pretty specific because the dogs are pretty accurate. But I want to know if you know, Yersinia or Pasteurella or any of these other respiratory pathogens smell the same to the dogs. Are they going to tell us that this animal's just sick? Or are they going to tell us, you know, is a sick animal smell like the same as a respiratory infection that's not MOV? Or is it different from MOV itself? Is that a very specific one? So, and as you can see, it's dirty. It's dirty out there. So these are all the things I've already mentioned to you. You know, these are the things that I'm worried about that we need to resolve. But what I want to keep going on here, and I'll tell you how we've been doing this work for about 3 years now. One of the things that I've realized is a lot of people, particularly when they're in a regulatory agency, they get very uncomfortable about this. And I always say, don't worry. You know, we're not trying to put Waddle out of business. We're not trying to you know, rewrite laws or anything like that. You know, we can use the dogs any way that a manager or a scientist or anybody wants to do it. And some of that might be to trigger testing, to say it's time to test for something. That may be as much as you want to do, is, you know, from an ecological point of view, or is this disease around, is it here, and other action may not be taken on it. But, you know, lots of people will sample animals at a you know, on a schedule. Well, maybe the disease is going to recur before then, and a dog might help you know when that's the time to go out and spend the money to catch the animals and then send them to you know, a proper diagnostic lab. The other thing that we're hearing from people is that sometimes they've just got a bazillion samples. This happened during COVID, right? There were thousands and thousands of the wastewater samples. And they said, maybe we ought to just use the dogs and see which ones they alert on, and then we send those in first. And then the other ones get sent in after, right? These are the highest priority sort of samples. It'd be a great use for the dogs. If you really believe in them and you think they're doing well, then maybe what you do is just prioritize and send in the ones that the dogs alert to, and then you subsample the rest just to see if they're missing them or they're not accurate or whatever. And if you really drink the Kool-Aid and you believe me and the dogs are great, then forget about the lab. But that's a little ways off. That's going to take a while. But there are resource limited parts of the world. Steve and I, you know, both have spent a lot of our time stomping around Africa where there is no Waddle. There is no other, you know, the dog may be the only thing that we can possibly sort of do. So that could be a realistic scenario. So we're going to keep working on this. We're going to do some of the aging experiments. We may also do them with bison, where we see if that odor signature declines or goes away over time. We're going to have the dogs at captures. We do them. We put the dogs and the olfactometers in a trailer so that they can work indoors, so that if it's windy or cold or whatever, they can do it and work. We're going to continue to refine that work, and then keep working on the swabs. And then we work with Kate. Kate's doing, she calls it the Big Mac study of biomeme and LAMP and you know, the field sort of methods. We're going to participate in that. And what I really want to emphasize is that the dogs are, they're just one of the tools in the toolbox here. I often say I have to find the money to feed a whole bunch of dogs, but I don't own anything, manage anything, have authority over anything. All of this conservation happens under the authority of other people. So we're here to present the tools for how a manager or a decision maker is going to use them. And quite honestly, that's, again, back to my original starting point. That's why I come to places like Cornell, is to say, how should we be doing this, and what's the best way we can do it that's the most effective? And of course, we're going to keep working on the spatial separation stuff, which, quite honestly, might be the most important intervention of all of this, is to keep them away and keep that transmission from happening. So, thank you guys. I wanted to leave a little time to actually, if you guys have questions, but also to hear feedback, because like I always say, I'd rather hear it from my friends, and I include all of you in that now, than get blindsided by something sort of later on. And tomorrow night I'll talk more about the general work, our ecological work, and our environmental justice, and all of that sort of stuff. But there are my coordinates. Steve and Casey have them too if you want to reach out to me later on. So thank you guys.